Welcome everyone to first ever video of my channel. Hope you all are safe and healthy. So today I am going to talk about Sanger sequencing. Now Sanger sequencing invented in 1977 by Frederick Sanger. It is not the first sequencing technique, it is the second sequencing technique, but it is the first SBS technique. Now what does means by the SBS? SBS means sequence by synthesis. Now it means we are going to sequence our desired stretch of the DNA by synthesizing its complementary strand. Now there are some main components we need to remember. So first one is the 2 prime comma 3 prime dideoxyribonucleotide presented as a DDNTPs, and it's act as a terminator. Now what that means by terminator? That means when the DDNTPs got incorporated in a synthesizing chain, it's going to terminate the reaction or stop the reaction. Now second one is the DNTPs. As we all know, DNTPs is required in any synthesis reactions. And third one is the DNA polymerase. Now remember, here we use only the Crenau fragment because it's lost the 5 prime to 3 prime exonuclease activity. Now, what is the difference between the DDNTPs and the DNTPs? Now here I draw the structure of the DDNTPs and DNTPs. Now as you can see that in DNTPs there is a 3 prime OH group that present, but in DDNTPs instead of OH there is a hydrogen in 3 prime. Now as we know, for synthesizing any strand or for incorporation in incoming DNTPs or bases, we need the 3 prime OH group, because as we see here, the 3 prime OH group, there is a lone pair in the oxygen atom. This lone pair going to attack the alpha phosphate of incoming bases or DNTPs, and the pyrophosphate get released. PPI gets released. So if instead of OH there is H, that means there is no lone pair, that means there is no attack. So no further base incorporations and the synthesis going to terminate it. Now let's see how these reactions or Sanger sequencing works. Now in Sanger sequencing we take 4 tubes. In 4 tubes we have our 4 DNTPs in same concentration, and we have DNA polymers and our template. Now the fun factor is in 4 tube, in each tube we have only one DDNTPs, either DDATPs, DDCTPs, or DDTTPs and DDGTPs, and the concentration of the DDNTPs with respect to DNTPs is very low. As an example, DATPs. If the concentration of the DATPs and the DDATPs is same, then there will be a problem in synthesizing the complementary strand, because as we can see here in our desired strand there is a 3 T's which complementary is A, so DATPs or DDATPs gets incorporated. If the concentration of the DATPs and the DDATPs is same, so the probability of incorporations of DATPs or DDATPs in this 3 position is same. So if we run, let's say 50 cycle, then out of 50 cycle, 25 times elongations of the complementary chain or synthesis of the complementary chain get terminated in this place. So the elongation process gets hampered. Now let's see the further steps of the Sanger sequencing. And after the mixing, we give the incubation as we want. After each incubation there are so much partially produced DNA strand. Remember, these all are double stranded DNAs. In next step we have to denature it to run in gel electrophoresis. So we denatured by heat we denatured by heat, and these are the single stranded DNA. And the further step is gel electrophoresis. In gel electrophoresis, we make four wells. Why four wells? Because we have four tubes, mixing tubes. In each well, we unloaded each tubes. So here we unloaded the tubes containing DATPs. Now what that means? It means in that tube which contains the DDATP, every nucleotide or every complementary strand which synthesized, the last base is A, because after incorporations of the DDATPs it's get terminated. Now in electrophoresis we use 7 molar urea and 70 degrees centigrade. Why 7 molar urea and 70 degrees centigrade? Because as we run the single stranded DNAs, so there is a chance or intention of the DNAs to form the secondary structure, and the 7 molar urea and 70 degrees centigrade inhibit the tendency to form the secondary structures. Now, as we know, in electrophoresis, molecules or strand gets separated, DNA strand gets separated due to its length. In electrophoresis, as we know, the shortest stretch of the DNA or shortest length of the DNA run the greater distance. In here, this is run the largest distance. So this one is the shortest. That means this strand get terminated fast. Now in complementary strand, this base, that is thymine was the starting base. Now from these we can determine what is the sequence in our complementary strand. Remember, this one is the gel picture of our complementary strand bases. Now in complementary strand, the sequence of complementary strand synthesized from 5 prime, 5 in 5 prime, first one is the T, then comes A, then comes C, then comes T, then comes A, again A. So 2 A's, then comes C, then comes G, then comes T, 3 prime. Now what is our interested DNA sequence or the template sequence? Template sequence run from 5 prime, A, C, G, T, T, A, G, T, A, 3 prime. So this one is the sequence of our template. Now see, we take this as our template. Now compare these two sequence is same. So for today that's it. Hope you guys enjoy it. Feel free to criticize. If you have any question, let me know in the comment box below. If you like my video, please click the thumbs up button. I thought upload a series of the sequencing videos, so subscribe for upcoming videos. Thank you for listening.